So if we want to talk about cell division of the eukaryotic cell, we have to think about what makes a eukaryotic cell different from a prokaryote. Why can't eukaryotic cells just cut themselves in half as easily as we saw with prokaryotes in cell division or binary fission? Most of the time we look at a much more complex cell. Remember, in prokaryotes, there are no organelles. Eukaryotes are going to have to deal with a nucleus. They're going to have to deal with organelles, multiple chromosomes, many proteins, and other macromolecules. There's a lot more going on, okay? So when we look at the process of dividing a eukaryotic cell, it's going to make it not only different, but a little more complex than we saw in our prokaryotes. Let's start with the chromosomes. When we talk about chromosomes inside your nucleus of a eukaryotic cell, we look at the idea that this chromosome is really chromatin. How is that any different? Well, when we talk about chromosomes, we talk about just DNA. When we talk about chromatin, we're actually talking about the DNA and the proteins. Why are we worried about this? The idea with the proteins is they're actually going to help us maintain structure and control the function during cell division. So during DNA replication, and then eventually cell division. So this is an important characteristic because we have to have these proteins to be able to go through this process. And we'll show you how we go through this entire process, but the image on your screen is showing you the chromatin beginning to condense. So see how it's all kind of fluffy around this outside? This is what we call the loose state of chromatin. It's all over the place. Reminds me a lot of cotton candy. And then as the chromosomes begin to condense, we're getting ready to go through cell division. So they have to condense down and become very, very compact and structured and organized in order to divide. Well, why? We want our chromosomes to actually look like the image on the left here when we go through this process. So we take a chromosome and we're going to go through replication and make what we call sister chromatids. Okay? Sisters are perfect copies. So it's one chromosome. We make a perfect copy of it. It's just like you putting it in the photocopier. And then we're going to split them out again. One goes to one cell, one goes to another cell. Now, for you, this looks fairly simple, I suppose. It takes about an hour in your cells to actually go through this process. Now, another term on here that you need to keep track of is something known as a centromere. You see this kind of spot or waste squished in part in the middle here. These act like little magnets to hold the sisters together. This is important because until we go through the proper stages of cell division, we actually need them to stay close together so that they do not get separated prematurely. What we will look at is how these two copies of these sister chromatids that are held together at that centromere will allow or will be divided during cell division. And when that happens, those sister chromatids will each one becomes a new chromosome in a new cell. You'll see how this goes as we go through mitosis. The idea with mitosis is we take those two sisters and we divide them into two new cells, but because they're identical twin sister chromatids, basically, we look at producing identical cells. This is very, very important. And when I say identical, I mean genetically identical cells. This is really important because it's what differentiates mitosis from meiosis for us. So when we're trying to get organized to do cell division, as we mentioned, our chromatid in our nucleus, they're going to have to wrap down, and this chromatin is going to fold and become very condensed and be in this sister chromatid state. And when it's in this duplicated chromatid state, when you see the chromosomes in the nucleus in this sort of X fashion, these are not X chromosomes. This is what the chromosomes actually look like for all of your 46 chromosomes that you have. When they're ready, and when they look like this, they're ready to go through cell division. How do you get very unwieldy, cotton candy looking DNA down into a nice tight package like this? Well, what we're actually going to do is take that DNA and wrap it around a protein. That's that chromatin we were talking about earlier. The chromatin adds structure and function and allows the DNA to go through this process effectively and correctly. And when we continue to roll and fold, and you see as we go along, it's condensing and condensing and twisting until you get essentially what we refer to as beads on a string. So those nucleosomes are these little green proteins that are going to wrap the DNA around them so that they actually have a good connection with those histone proteins that we were talking about before. And we just keep wrapping and wrapping and wrapping and wrapping until, and you can see it occurring in an SEM here, we just keep wrapping and wrapping until we get to that nice condensed state we were talking about before. So if this is DNA plus proteins, where are the genes? Well, remember, genes are sections or pieces of our chromosomes, right? So these strings of DNA that you can see are actually genes. And we'll look at what that means later on. So for you, we've got to keep in mind for every species, the number of chromosomes that you have in your nucleus is species specific. So for humans, you have 46. We talk about 23 pairs or 22 pairs plus XX or XY for female, XX for male, XY. So when we look at these, you can see you may not consider your dog to be more complex than you, though your dog has 78 chromosomes compared to your 46. Does that mean your dog's more complex? No. That has nothing to do with it. We look at the distribution of genes and genetic information on chromosomes. So we have to think about how this is just an organizational pattern for the genetics. But what we look at with your 46 chromosomes is we actually look at them, think about them like shoes. So you have pairs of chromosomes or pairs of shoes. And 22 of those pairs, you get one from your mom, one set. So of these, you get 22 chromosomes from mom. And you get 22 chromosomes from dad. And then you've got two more we got to deal with. Well, you get an X from mom. Almost always. because mom is XX to be female, right? So she can give one X and one X. That's it. Dad, however, is XY. So he could give you an X or a Y. So this is where the sex of the baby is determined. Does dad give an X or a Y to the offspring? If he gives a Y, the offspring is male. If he gives an X, the offspring is female. So these pairs are really important, and we'll get into this more in DNA and RNA and genetics later on. Homologous chromosomes, we look at these as being not perfectly identical. These are not sisters. Sisters, perfectly identical copies, right? We saw that. Sister chromatids are made during DNA replication. Homologous chromosomes, you get one set from mom and one set from dad. So this, putting them together, gets a fertilization event or reproduction. So very different purpose. So what else is different about them? If these are not perfectly identical, so take your gene for hair color. It's carried on a chromosome. Let's say chromosome 7. It doesn't matter. It's not actually 7, but just for reference. So you get a chromosome 7 from mom. You get a chromosome 7 from dad. Maybe mom says you're going to have brown hair. Dad says you're going to have blonde hair. They both are coding information for hair color, but mom's is a different variety than dad's. Mom says brown hair, dad says blonde hair. We'll look later in genetics who actually wins this competition, so to speak, when we put these two together and get you. But you have to keep in mind that with these homologous chromosomes, we're looking at chromosomes that have the same coding genes, but they might be what we call alleles, or variations of a gene. So we haven't mentioned two things on here yet. Karyotype, we'll get into this later. This is essentially a picture of your chromosomes. And we've got to start talking about what this interphase and cell division process mean in mitosis.